Hi students, good evening everyone, very much good evening. Very slowly everyone joins in. So we wait for another 5 minutes and then we continue for the what is this cable for? Just a second, I have so many cables down here. Superb. So in meantime, what books are you reading in your house? Which books are you making readable beyond your textbooks? Are you reading any other books also? Let's just discuss some books contents that I'm going through at the moment. That's the what I'm speaking here. It's coming maybe 2 minutes or not 2 minutes, 10 seconds or 30 seconds later. So this is book called The Power of Habit. Uh, it's by Charles Duhigg. So it has very nice chapters actually. If you got the chance to read this, it can change really different personality. It will tell us the habit loop, that why we do one thing again and again, same thing. A bit psychology of our human brain. What should be the keystone habits? The power of crisis, neurology of free will. I mean, all these chapters, they make really good sense, such as the power of brushing our teeth in the morning. How does it came? So there was no one was doing brush, but we have this habit now. So it's all about habits in our life. So that's the most common one. The 7 Habits of Highly Effective People by Stephen R. Covey. The most oldest and most well-known books. More than 25 million copies have been sold for that. So this is also must read. Um, in order to, I mean, I'm sharing these book details today is just to make sure that you should not be just around your textbooks or your college examinations. Go and beyond your own, you know, your beyond your own level of sphere you are, or I am actually, whosoever where we are at the moment. So we have some specific place, we have some specific, you know, yeah, Nidib Joshi is one of the best books, right? So we are all in the zone of one place that we think we are best known for us, but there are always chances to grow. There are always chances to be more. There are always chances to be bigger. There are always chances to be wiser. There's always chances to be more calmer. There are always chances to be more healthier. There are always chances to be more wealthier. There are always chances to be more good position in the society. There's always things are always available for us. It's just we need to identify our inside, and that's all things. That's how can we grow. It's all present in our inside. It's not outside or not somewhere else. It's been always been inside. We have to discover it, how to get better and how to get bigger. I have shared one this Ikagai with you. That's actually a life-changing book for me. This makes me to think who am I, what's my passion is, how can I be a better person, how can I be do what I like, what how could I make my passion, what the world needs, what could be paid for, what I love. So bringing my passion, mission, profession, and vocation, and in order to work my Ikagai. So this is also very nice. Yeah. And I don't know if you are spiritual, no. So some are spiritual, some are materialistic. It's depends upon how you are looking your life. For the spiritual side, by Jay Shetty, he explained his monk side of his life according to Krishna's temple, ISKCON temple, Hare Krishna. He was there, and he just explained his life there, how his life got changed. Born and brought up in UK, wanted to have UK style lifestyle, but suddenly coming to India, living with the monks, living on the very less very less input. You know, there's nothing required. You just sleep on the floor. You just need two-time foods, simple food, water, um, meditation, lots of meditation, chanting the God's name, and then yeah. So that's the something about it. So we will share more titles in future. Yeah, 5 a.m. That's that's if I could make that thing happen in my life, the 5 a.m. rule, I will be the most happiest person. To wake up at 5 a.m., do your regular exercise, make sure you do some meditation or something like concentration, and then plan your day for the whole day to win over it, and start working by 7 a.m. or 8 a.m. And yeah. Okay, I hope most of you have joined now. So we come to another part, that is the education part. So this was moreover like so just main reason to show you these things was keep a balance, not just the textbooks but also some self-help books. There's nothing wrong in reading them. Some might say, oh, who is that reading those self books again? But believe me, if you can improve, you can be a better person of yourself. There's no harm reading them. There's no harm in reading them. Okay, I put my stacks there again, and then we start with the. So yesterday we were discussing about basics of molecular biology. In order to understand, we discuss all the structure of DNA, RNA, transcription and translation. We went with many animations yesterday. So today our focus is to continue with that part in the presentation form and discuss those other leftover RNAs that we left yesterday. So transfer RNA, tRNA, which is a small RNA chain of about 80 nucleotides that transfers specific amino acids to growing polypeptide chain at the ribosomal site of protein synthesis during the translation. Yeah, so it's helping, as we discussed in the last animation, that your DNA is coming up. It's making your mRNA, then mRNA is going to cytoplasm, and then from the cytoplasm, with the help of your ribosomal RNA, this tRNA is making various amino acids polypeptide chain and continuing the site. So it has sites for amino acids attachment and anticodon regions for codon recognition, and that site binds to specific sequence on the messenger RNA chain through the hydrogen bonding. Then ribosomal RNA, so this is of two types, 70S and 80S. 70S is made up of 50S and 30S, and 80S is made up of 60S and 40S. Yeah. So ribosomal RNA is the catalytic component of the ribosomes, and eukaryotic ribosomes contain 4 different ribosomal RNA molecules, 18S, 5.8, and so on. So main difference between your RNA and DNA is RNA is RNA nucleotide contain ribose sugar, and DNA contains deoxyribose. And RNA has a base uracil and DNA has base thymine. And RNA has presence of 2 hydroxyl group as 2 position of the ribose sugar, and this lacks of hydroxyl group at the second position of the ribose sugar. RNA is usually single stranded and DNA is usually double stranded. So proteins, now we have discussed our DNA and RNA. Now comes to proteins, which is also known as polypeptides, are made up of amino acids arranged in a linear chain and folded into the globular form. So the sequence of amino acids in a protein is defined by the sequence of a gene which is encoded in the genetic code. The genetic code specifies 20 standard amino acids. So this is the amino acid chain. Here we can see. Yeah. So together, so here the 2 cysteines, they are making disulfide bridges. This is leucine, arginine, cysteine, glycine, arginine, isoleucine, trypsin, and so on. And then they are making this tertiary structure, and these tertiary structures could be found out in pulses, peanuts, kidney beans, fish, chickpea, red meat, eggs, chicken breast. So all are the various sources of your protein. So all the people who are going for gym or making their muscles, so they tend to have more protein in their diet rather than carbohydrates and fats. So these basic players that we started yesterday, DNA and RNA protein, so how they are correlated with each other. So DNA could go replication itself, and then with the help of transcription it could make RNA. Then after making a transcription, RNA will make protein, that is translation. And this is the central dogma of life. Then DNA replication. So DNA replication is the basis for biological inheritance, is a fundamental process occurring in all living organisms to copy their DNA. So in the process of replication, each strand of the original double-stranded DNA molecule serve as a template for the reproduction of the complementary strand. So 2 identical DNA molecules have been produced from a single double-stranded DNA molecule. So here we can see during your DNA replication, you have your mother or you can parent DNA. So with the help of topoisomerase enzyme, you uncoil your DNA, and then helicase enzyme will unwind it, like it will make 2 parts of your DNA. Then their single-stranded binding proteins, they will come and join it there to make the DNA strand to be more stable. So it will make 2 strands, leading strands and lagging strand. So the leading strand will actually move from, it will make the opposite strand from 3' to 5' end. So leading strand, it will have the Okazaki fragments. Yeah. So you can see this. So it will have opposite ends, and then so this will make, with the help of DNA polymerase, it will start making the corresponding nucleotide base pairs, and hence it will go very smoothly without any obstructions. On the other hand, there will be a lagging strand here, which will be also being made with the help of DNA polymerase. But sometimes there will be segments. These segments are called Okazaki fragments, are named by the famous Japanese scientist who found them. And they could be healed by the DNA ligase, joined together by the DNA ligase enzyme. And here also, in order to make things correct, your DNA primase and RNA primer will work out to make things done. So after replication, there is a transcription. As we know, it's a process of creating an equivalent RNA copy of sequence of DNA, and transcription is the first step leading to the gene expression. So DNA gives rise to transcription, that is RNA, and hence then reverse and then this RNA could make also reverse transcription. So during transcription, a DNA sequence is read by RNA polymerase, which produces complementary anti-parallel RNA strand. And transcription results in RNA complement that includes uracil instead of thymine. Then transcription process, the stretch of DNA transcribed into RNA molecule is called transcription unit and codes at least one gene. So if the gene transcribed encodes for a protein, the result of transcription is messenger RNA. And this messenger RNA will be used to create that protein via the process of translation. So alternatively, the transcribed gene may be encoded either ribosomal RNA or tRNA, other components of the protein assembly process or other ribosomes. And DNA transcription unit encoding for protein and regulatory sequences that direct and regulate the synthesis of the protein. So here we can see DNA is read from 3' to 5' end during the transcription. So here you have your DNA polymerase, which is a self-complementary sequence. So only one of the two DNA strands called the template strand is used for transcription because RNA is single-stranded. Another strand is called the coding strand. So at the ends, you will see a single strand and then double strand with a hairpin loop, your mRNA is being produced. And also here we can see the chromosomal DNA with exon 1, exon 2, exon 3 with all the genes present in them with intron 1 and intron 2. Yeah? And then transcription, RNA synthesis with the help of RNA synthesis, exon 1, 2 and 3. Your introns will be removed which are not coding regions, and mainly the coding regions will be there, exon 1, 2 and 3 for your messenger RNA. So also the reverse transcription, which is a reverse transcribing viruses, replicate the genomes by reverse transcribing the DNA copies from their RNA, and these DNA copies are then transcribed to RNA. So retrotransposons also spread by copying DNA and RNA from one another. So here we can see that the HIV RNA with the help of reverse transcriptase enzyme, it's producing this double-stranded HIV DNA in these cells or cytoplasm. So this process is called as reverse transcription. So in general, again a summary of a central dogma. It's coming again and again because I'm repeating this because I was an interview at the Government Institute of India, one of the highest, one of the well-renowned institute. It was Indian Agriculture Research Institute, and they asked me central dogma of life. After doing my PhD, after studying so much, and suddenly they asked the first question: tell me the central dogma of life. This happens when you have studied so much within your own field, but you are not aware of the basics. Suddenly somebody asks you about the basics, and so that was my weakness at that point. I was so much studied about my own topic that I forgot all the previous studies that I did in my bachelor's and master's. Not forgot, it's just I didn't review them or didn't made a click for myself. So the mistakes I did in my life, I don't want anyone to do that again. So make sure that all these presentations that I'm sharing with you, make sure you have always with them and before any interview or anything, just have a quick review about them, have a quick read about them. And for the same, that's why I told write some assignment about them. So like a complete summary about whatever we do in the classes. So this will be your self-made notes, which you can use it in the future anytime. Yeah, some students, they have very sharp memory, they can remember things. Someone like me, I have no sharp memory, I have to admit that. I tend to need to things write down with me myself. I don't have that hard disk in my brain that I can save lot of data. Yeah, so there are weaknesses in your life, and we have to admit them. And in order to do so, once you have admit them, you find a way of solution of it, irrespective of being egoistic. No, I'm fine, it's not with, I have no problem with me, I'm all fine. So that's thing we need to yeah, remove that curtain and then agree the things that there is something and how we can recover that and find a solution out of it. Yeah, so there is always a solution, it's just we need to admit ourselves. Yeah, yeah, Shrutika. So central dogma is all about making your DNA. Yeah, sure, I will share the pictures also of them. No worries, no worries. Actually, buy paper books. I mean, e-books are quite hard to read, but nowadays there is audio books are also very popular. But in order to understand these audiobooks, we need to be have very nice hearing capabilities to hear those accents. They could be also Indian accents for the audiobooks, but you need to have very sharp. We tend to listen or to see things like English sitcoms over the television. But in order to listen to an audiobook is also one of the great practice, and it saves a lot of time also. It's up to you. Someone likes audiobooks, someone like to read paper books, someone likes to have Kindle ebooks. So it's everyone's their own preference how they wish for. So that's I was explaining my personal journey with the central dogma. That's the I'm explaining the central dogma here, which made me to lose my actually interview, and not lose because it was an agricultural interview, and I had my previous work in the cardiovascular physiology in the cardiac department. So there was a mismatch of the yeah, it was a government job, very highly paid. I just thought maybe I could get in, but no, I couldn't. So things happen for the good. Um, yeah. So DNA here is a general thing with the self-replication of DNA, and then you can have an RNA out of it, and then from RNA you can have a protein. And during this process, from DNA to RNA, it's called transcription, and from RNA to protein, it's called translation. RNA could also do self-replication, same as DNA. That's a special case, and also RNA could make go to DNA to make reverse transcription. Only these processes, students, only these processes are possible. It's not possible that DNA could make directly to protein, or protein could make RNA. So no way, no way. These things are not possible. Yeah. So or protein is making DNA. This is also not possible. So now translation, which is the first stage of protein biosynthesis. In translation, messenger RNA produced by transcription is decoded by ribosomes to produce specific amino acid chains or a polypeptide that will later fold into an active protein. So translation occurs in cell cytoplasm where the large and small subunits of the ribosomes are located and bind to the messenger RNA. Then translation process, the ribosome facilitates decoding by inducing the binding of tRNA with complementary anticodon sequence to messenger RNA. चाय वो नींबू वाली बिना दूध पे आंधी है लेमन टी जो दे दो दूध वाली चाह दे दो ग्रीन टी दे दो okay. The ribosome facilitates decoding. That's the translation that we have discussed also yesterday in the class. So you have a messenger RNA and this is a ribosomal RNA. On top your tRNA comes here with these three anticodons. So you have valine, alanine, glycine on the messenger RNA. So these codons for these amino acids, right? So tRNA will come corresponding to that and with the amino acid attached to them. So in this way, your protein chains of amino acids like methionine, glycine, leucine, valine, alanine, and so on. So next is glycine is coming, then phenylalanine is coming, and its chain is keep going on. So that's how at the end these polypeptide chains of amino acids are being produced. Yeah. My voice is not coming, students, or is fine, I think. Maybe check, turn off your Voice is not fine. My God, what happened? Maybe try to turn it off, turn it on. That's the only solution I can give. Um, Turn it off, turn it on. Oh, I haven't shared with you the, I think, Wilson and Walker, isn't it? Where is Wilson and Walker? Yeah, that's good point you made here. So I have this book of the, that's the best book that you brought a very nice point here. I do Fabrica, MBBT. So I'm sharing this PDF, the ebook of Wilson and Walker. That will be very, very beneficial actually. I actually made this content of your sessions here based from the Wilson and Walker only. There is nothing I took from anywhere else. I just read through the contents of Wilson and Walker and made this this 10 days workshop. It's the one of the best I would say so far they made this book. Even I want to write a book actually on the same, so I'm planning on it as soon as I have some times. Time is the key here. I will also write one book about it. So time is the key, students. You are at the moment most enjoyable and most happiest and most stress free part of. It's noisy. Maybe my laptop is very old. It's 8 no, in 2013 I bought it at the start of my PhD. So this is 2021. So 7 8 years. Yeah, let's see if I could adjust a new one. Sorry for that, the audible. But I hope, but for most of the time, most of you, audio will be fine, I guess. Because I've joined with the mic now. Because with the mic there is a clearing, voice cutting, clearing. So it's filter out that thing. It will be fine. Okay, so yeah, as I said, so far Wilson Walker is the best book in the case, right? Wilson Walker, and beyond that, you can, yeah, I can share some more books also. So let's say from the biochemistry, from this also you can have some techniques. I have from Lehinger, Lipman-Cord, and Harper, three one of the best books ever for the biochemistry. So this could be also beneficial for you from the study perspective. Please don't just let these books to be just for like in there. Do read them. Wherever you have any questions, instead of finding them on the Wikipedia or over the YouTube, try to find these answers in these books. It has accurate one of the most accurate information that you can get it. Yeah, so these are your Wikipedia and Google actually. So whichever information you are looking for, go and read from them only. So this is taking some time to get upload. So we can continue with our lecture now. Okay, the translation is over. So yeah, I was talking about many amino acids so far, phenylalanine, serine. You might be thinking that there are so many names. So this is the full forms or the genetic codes of these names. That is UCAG. This is the first base. UCAG. You write on this pattern, and then you write UCA and G. Yeah, and on the right pattern you write U C A G. Then again U C A G. Then U C A G. U C A G. So what you do is, for the first base pair you take it from here U, then second is from here U, and third from here U. Then again U U C, U U A, U U G. So this will be your phenylalanine, and this will be your leucine. This will be your serine, and so on. So most important are the start codon, that is AUG, methionine, your stop codons UAA, UAG, UGA, these three ones. And then you can remember these together. First you start to remember the one with the four in a box: leucine, proline, valine, alanine, threonine, glycine, arginine. Yeah. Then try to remember these two ones with that group in two: serine, arginine, aspartic acid, lysine, asparginine, glutamine, histidine, and yeah, isoleucine, these three together. So what is genome? So we are done with this translation, transcription, central dogma, DNA, RNA, all the proteins, all the basics behind it. So now let's discuss about genome. Now genome is entirety of an organism hereditary information. It tells the whole organism hereditary information. It is encoded either in DNA or many types of viruses in RNA, and the genome includes both the genes and the non-coding sequence of the DNA. So comparative genome sizes of organisms here is written here. So we can see this Homo sapiens, Musmusalus, Drosophila melongster, Rhabdopsis thaliana, roundworm, C. elegans, Saccharomyces cerevisiae, Escherichia, and Haemophilus influenza. So E. coli and bacteria, they are one of the best organism to study about prokaryotes. And for the eukaryotes, if you want to study any patterns in human diseases, so mouse is used for the genetics Drosophila. In the plants we use Rhabdopsis thaliana. And to study more about human diseases at the smaller level, C. elegans and Saccharomyces, they are one of the best patterns so far to study about it. Yeah. So why they have been chosen, why these patterns, these organisms have been chosen for the genome studies. It's basically, you can see the size base pair, the comparison is written in there. One of the important tables here. So human sapiens have 3.2 billion base pairs, and chromosome number is around 46, and the mouse is, the chromosome number is around 40 and 2.6 billion size base pair. So almost similar. The configuration seems quite similar. Whereas if you want to study about genetics, it's 137 million with the 8 chromosome. For the genetics is one of the best. In the plants, this is 100 million with the 10 base pairs. And for the prokaryotes, this is only 1 and 1 chromosome number with 4.6 and 1.8 million size base pairs respectively. So why genome analysis? So the prediction of genes in uncharacterized genomic sequences to obtain complete sequence of as many as genomes as possible, for genetic modifications, to develop new varieties at a faster rate like BT cotton and BT brinjal. So tools that are used in molecular biology. So there is a gel electrophoresis. So the basic principle is that DNA, RNA, proteins can all be separated by means of an electric field. So now we're gonna discuss about a little bit, little bit about all the tools in molecular biology, but not in details. But we will do them in our upcoming sessions. So if we are studying about basics of molecular biology at the moment, so I thought, why not we touch also some tools that are used in the molecular biology. Yeah. I mean, Pooja, at your age, if you can remember these genetic codes, which codons code for which amino acids, it will be very beneficial for you if you want to be non-study about proteomics or genetics or any field you want to go, because these will be on your tips, right? Because you know what happened, I'm telling you a psychology. At your age, at very tender age of your, like you are maybe 24 23 years if you're in bachelor's, or 27 if you're in master's, 25, your brain tends to absorb many things as fast as possible. But as the age increases by 30 plus, I'm 34 at the moment, you can learn many things, but by the experiences and everything, our brain starts to function, do multiple things, yeah, can do many things, but the tendency to remember the sharpness that we tend to have as a younger age, of like when I remember when I gave my plus 2 biology exam, I have to remember all the physiology of human body, all the physiology of plant metabolism, and it was a different level when I did that. But once I went for my masters or one for my PhD, the level of my brain, the maturity of your brain, the experiences that you had, it's different. So I would suggest at younger age, the more things you can do, the more certificates, the more experiences, the more learning you can make. Yeah, it's always beneficial for you. So because by the end of age, things will come over. There will be many things will coming up. But at this stage, you can learn a lot. That's the best time. That's the best time. Cool. So let's, sorry for that line. So gel electrophores, the basic principle is that the DNA, RNA and proteins can be separated by means of electric field. So in agarose gel electrophores, DNA and RNA can be separated on the basis size of running the DNA through an agarose gel. So protein can be separated on the basis of size by using an SDS page gel, or on the basis of size and electric charge. So you have a negative electrode here and a positive electrode. So negative and positive, they are both on the opposite sides with electric current going through them, and DNA bands are separated by size here. Yeah. So polymerase chain reaction. So polymerase chain reaction is extremely versatile technique for copying DNA. So PCR allows a single DNA sequence to be copied or altered in the predetermined ways. So PCR has many variations like reverse transcription PCR for amplifications of RNA, and real-time PCRs which allow for the quantitative measurement of DNA or RNA molecules. So in this PCR analysis, I want to, on the basis of this PCR, there is a separate presentation. So yeah, so PCR, it's a technique which takes specific sequence of a DNA of a small amount and amplifies it to be used for the further testing, that is in vitro technique. So as we see the short history of PCR, in 1983, Dr. Kerry Mullis developed PCR. In 85, first publication of PCR by Cetus Corporation, which appears in Science. Then in 86, purified Taq polymerase is first used in PCR. And then in 88, Perkin-Elmer introduces the automated thermocycler. Then in 89, Science declares Taq polymerase, that is molecule of the year. The purpose is to amplify a lot of double-stranded DNA molecules with the same size and sequences by enzymatic method and cyclic conditions. So in the denaturation, that is a DAE method. So in DAE, first is a denaturation at temperature from 92 to 94. Your double-stranded DNA melts to the single-stranded DNA at 92 degrees Celsius. So they got all separated. Then annealing is happened around 50 to 70 degrees Celsius. Primers, forward and reverse primers, they bind to the complementary sequences. Then the extension, that is temperature of 72 degrees Celsius, time is 0.5 to 3 minutes, and DNA polymerase binds to the annealed primers and extends DNA at the 3' end of the chain. 5' Taq polymerase and 3' Taq polymerase and so on. So there's products of extension, yeah. Then overall principle of PCR, then DNA one copy, and this is the PCR copies here. Then chemical components, it includes magnesium chloride 5 to 2.5 millimolar, and buffer's pH is 8.3 to 8.88. Then DNTP is 20 to 200 micrometer, and primer 0.1 to 0.5 micrometer, and DNA polymerase 1 to 2.5 units, and target DNA less than 1 microgram. So basic requirements, you have to need to know the DNA sequence of target region that must be known, and then primers that are typically 20 to 30 base pairs in size. Then thermostable DNA polymerase is an example like TARC polymerase, which is not inactivated by the heating to 95 degrees Celsius. So DNA thermal cycler machine which can be programmed to carry out heating and cooling of samples and number of cycles. Then applications of PCR, molecular identification, sequencing and genetic engineering and so on. So this is the instrumentations that we used, that is here, there are 4 types. This is multiple polymerase you can do. This is light cycler. This is the most old one, but this is the most common one that we use in the PCR machine. Yeah. So that was about PCR. Okay. So now we will check how PCR is done in the lab, but before it does, I need to make sure that these are the books that I have shared with you, they being uploaded interfere with the video. Okay. Genomic studies differ from metaproteomics. I mean, it's genomics, as we can see, it's more concerned with the genome part, right? It's dealing with the genome part here, but proteomics is all about, okay, there is some problem with the streaming here. There's a problem with streaming here. Just a second. That's the irritating part when you're having a smooth lecture and suddenly it start to lag. Just a second, it will take 1 or 2 minutes, it will be fine. Just a second. I hope it's fine now. Please let me know if everything is fine. My voice is coming over to you on time manner. I hope it's fine now. Just give it 20 seconds or 10 seconds. It is fine, I think. I can see on my window the things are fine at the moment. Because it doesn't make sense if I keep speaking from my side and you're not getting the information and we make the lectures. That's the most horrible part of online teaching. Suddenly lack of signals and things got abrupt. Is it fine, students? Please confirm. I think there's no lagging is happening now. Sounds good. So we have just discussed about PCR in our above part, right? So I'm gonna make now one video about this PCR, that how PCR is working. So yeah, this PCR analysis we have just discussed. You have a copies from 2 raised to power 2 copies, 2 raised to power 3 copies, 16 copies, 32 copies, and so on. By then you have 35 cycles, you have around 68 billion copies. So a primer is a strand of nucleic acids that serve as a starting point for DNA synthesis. So these primers are usually short, chemically synthesized oligonucleotides with a length of about 20 bases, and they are hybridized to a target DNA, which is then copied by the polymerase. The minimum primer length used in the most application is 18 nucleotides, and replication starts at 3' end of the primer and copies the opposite strand. So in most cases of natural DNA replication, the primer for DNA synthesis replication is a short strand of RNA. So some applications, this is the task for the DNA sequence can be assisted by PCR. Numerous applications for traditional process of DNA cloning. We can match the parental testing also. So let's check now, students, how this PCR is done. Yeah, so I will remove my mic now. Lady will explain this virtual lab about the protocol.
Before beginning this protocol, students must fill out the worksheet provided on the last page of this document. This will allow students to calculate the appropriate volume of reagents to mix in the creation of a master cocktail. The purpose of a cocktail is to mix all the common reagents in a multi-sample procedure to avoid excess pipetting and potential errors. In this example of Protocol 2A, 4 students are amplifying the actinin 3 gene from all 4 of their DNA samples. This information is filled out on the left-hand side of the worksheet. On the right-hand side, students use the equations to determine the number of tubes needed and the multiplier for reagent volumes. In addition to the 4 DNA samples, a negative control sample is always included. Therefore, 5 tubes are needed for the PCR reaction. In the creation of the cocktail, pipetting error is accounted for by adding another sample. Therefore, the multiplier for this group is 6. Each row of Table 1 represents a common reagent used in all the samples undergoing a PCR reaction. The multiplier is filled out in all rows of the multiplier column of Table 1. Moving across each row, the volume indicated per reaction is multiplied by the multiplier. (12.5 times 6 equals 75 microliters of red tack added to the cocktail. 11.5 times 6 equals 69 microliters of water added to the cocktail. 0.5 times 6 equals 3 microliters of each of the forward and reverse primer added to the cocktail. Finally, the sum of these volumes should be equal to the product of the final row. 75 plus 69 plus 3 plus 3 equals 150 microliters of total volume in the cocktail. This amount matches 25 times 6. Table 2 is used as a guide for labeling of the PCR samples. As indicated by the equations, this group will be using 5 tubes. The class must collectively number the PCR samples to not confuse samples amongst the groups. In this example, these students have been assigned samples numbered 5 through 9. The sample names are filled out in the second column, and the primers used for each sample are filled out in the last 2 columns. In this example, the forward primer is actinin 3F and the reverse primer is actinin 3R. The same primers are used for all samples. The common reagents provided are molecular biology water, a forward primer, which you should be sure to check for the marking of PCR to ensure that you are using the appropriate concentration, a reverse primer, and red tack ready mix. The red tack will need to be thawed prior to use, but once thawed, keep the solution on ice to prevent enzyme degradation. Next, prepare the cocktail by using the volumes and reagents indicated on the worksheet. Obtain a new tube and label it as the cocktail. When creating a cocktail, it is best to start with the largest volumes. Though red tack is the largest volume, the solution should be kept on ice as long as possible, so instead the water is added first. When adding each of the reagents, be sure to use the appropriate micropipetter for the volume being added. Next, the primers are added. It is best to pipette small volumes directly into larger volumes to avoid loss of the reagent. Finally, add the red tack and immediately return the stock to ice. Mix the solution and centrifuge all cocktail tubes created by all groups in the class together. and hold the short button to do a brief spin.
That was the part 1.
The collected cocktail solution will be split amongst the 5 tubes in this example. PCR tubes are provided in strips of 4, so if you need 5 tubes, use 2 strips of tubes. In this case, to amplify actinin 3, 24 microliters of cocktail solution is added to each of the 5 tubes. Before adding DNA to the PCR reactions, make sure that the tubes are labeled. Make sure all DNA samples are mixed before transferring the solution. One microliter is a very small volume, but you should still be able to see a drop in the tip. Put the tip all the way into the bottom of the tube and pump up and down on the plunger before expelling to the second stop. This helps to ensure that the 1 microliter of DNA has been expelled into the reaction. Cap the tubes tightly and flick to mix the samples. Use the mini microcentrifuge with the 8-tube strip head to collect the solution at the bottom of the tube. It is good practice to always mix and collect solutions before every step, especially if the solutions have been sitting for a while. Turn the thermocycler on with the switch in the back. Place the tubes in the thermocycler by pressing the metal latch and lifting up on the lid. Tubes can be placed in any spot or orientation. Close the lid and make sure the metal latch has clicked into place. Heat cycling programs have already been programmed into the thermocycler, so all you need to do is run them by pressing the Proceed button. Use the horizontal arrows to navigate to the program of interest, in this case ACTN, to amplify the actinin 3 locus, and hit Proceed. The heated lid should be enabled by hitting Proceed to prevent extreme condensation at the top of the tube. The thermocycler will ramp to the initial temperature and begin timing the incubation. It is good practice to make sure the machine reaches this step with no errors. After it has initiated properly, the machine can be left while the reaction proceeds for several hours. Once the run is complete, the cycler can be turned off by simply flipping the switch in the back of the machine.
Okay. Now the protocol is approved. It's all good now. We'll share this over the. Oh, low battery. That is not good. So you can check the protocol too within this link. Okay, I think you have a class now, next class. My battery is almost finished, 12%. It will go off now. So thank you very much for today's session. We have to just do this PCR today. Tomorrow we will do, we will make sure we will do the DNA isolation with this perfect protocol also. So in this way our classes will continue. And I will repeat one more time. I have to go for 1 week traveling with my brothers and sisters. I might be not available for the classes, and I might be beaten up by my superiors at the Edu so we need to find some solution so that these lectures should be delivered by me only, and it should not be the previous lectures. And I want these lectures to be live with you. Yeah. Because there is a difference between recorded lectures and live lectures. There is an interaction I am having with Vaishnavi, Ritika, Ramesh, Shrutika, Shipra, Soumi, Asima, Saujit, Soumi. So yeah, there are many students who are replying here. So you're all very important to me, and I want that the information that I have, I should deliver to the best possible way to all of you. And problem with me is lack of time. So please let me know some solutions to it. I beg you all. I don't know how, but I have to go for 1 week or so, more than 1 week, and I will be on regular traveling. And yeah, let's see. Maybe we can have in a single day 2 hours class. Yeah, in a single day, if we have 2 lectures for 2 hours, that will make things easy for me and for you all also. So we both could be benefited out of it. That's the best solution I could think at the moment. So in the group, we can discuss the timings where we can make 1 hour and 2 hour, like 5 p.m. in the class or somewhere in the morning, or maybe late night 9 p.m. I have no problem. If you have no problem, I can give a lectures at 9 p.m. also, 9 to 10. Yeah. So I will write one message if everyone agrees to that from 5 to 6 and 9 to 10, and also over the weekend. Yeah. Can you please reply at the moment there are 36 students in the class? Is it fine if I make class for you 5 to 6 and 9 to 10 also? Yeah, we will go then very slowly. We will not, because otherwise if I start to make things faster, you will not understand, and I will also not like as a teacher to you giving information. It has to be go slow and steady, and this way. So I can see can getting positive response for 9 to 10 also. Okay, good, good. So okay, we make 5 to 6 and then we make 9 to 10 also. Can we do it today also, 9 to 10? I'm free today also, because I can see around 50 students who are coming here in these classes, and the one who are coming are replying to these chats here. It's a humble request. If you are agree to that, I will write in a message. So you just all agree with the thumbs up in the group also, so that even my higher authorities, they say it's okay, it's no problem. Because yeah, there's management, there's trainee, there's students, and all should work uh, with hands in hands. Yeah, superb, nice. Thank you so much. So maybe I can make it. Oh, first I should share the PCR. So, and the today's lecture that I was giving, it is already being shared yesterday. So please make sure you are pinning them down so that you can come and check them anytime. Where is that? Yeah, this is Introduction to Basics. Pin them. It will be very beneficial for you. So like this. So no, this is already pinned. That's good. Awesome. This here is also there. So I just make sure. Just reply with yes to this. I will write it later. No worries. So thank you very much. Wish you all nice evening. Enjoy your evening then. Uh, so I will make sure that we also meet everyone at 9, because if somebody no, it will be from 9 to 10 only. After 10 no class, so that after having all the knowledge you can sleep very well. Believe me, it's act like a therapy. If somebody is having insomnia or something, just read molecular biology with a proper dose, you will get a really good sleep. Then see you again at 9 then. I will write a message in the group then. Yeah. So thank you very much. Wish you all nice evening. Bye bye.